Hi everyone, my name is Emily Opet and I chose maple syrup urine disease as the genetic disorder that I would be studying throughout the class. So I'm going to go over that with you in these next couple slides. So first, what is maple syrup urine disorder? Basically, it's an amino acid disorder, which means that your body cannot break down amino acids that typically come from the foods you eat, such as foods high in protein. Um, Another common amino acid disorder that some of you may have heard of is phenylketonuria. And so basically, in maple syrup urine disorder, there is a deficiency in the production of enzymes that break down three branched chains amino acids, which include leucine, isoleucine, and valine. And if this goes untreated, these three branched chain amino acids will build up in the blood and then can negatively affect your neurological systems and can eventually lead to death if not treated quickly. And this disorder was first discovered in 1954 by Dr. John Menkes, and he discovered the disorder after four infants from the same family all died within the first 3 months of their life. And while he was trying to treat them and figure out what was wrong with them, he discovered that their urine and their sweat smelled very sweet like maple syrup, although at the time he still did not know what the disorder was. He just thought it was some sort of neurological condition. So it wasn't until a couple years later that it was actually discovered and given its name. The prevalence, it's not a very common disease or disorder. It happens in nearly 1 in every 185,000 newborns worldwide. However, interestingly, this disorder is actually very common in the Old Order Mennonite population. It occurs in every 1 in 380 newborns. So compared to worldwide, that's actually very common. There isn't specific research and reasons why it's so common in this population. However, it's speculated that Old Order Mennonite people in that population probably reproduce with others that are in that population as well. And since it's a very small population and this is a genetic disorder, they probably just reproduce with people who are either have the disorder or are carriers for the disorder. The physical effects, there aren't really any physical effects that you would see on a newborn right after it's born. However, their urine will be very dark and representative of maple syrup. However, that's not common, or that's not the case in all infants with this disorder. Other common symptoms that you might see very quickly after birth do include lethargy, poor feeding, vomiting. The most common is the sweet smell in their urine and their sweat. And then, if not treated quickly, you might see neurological, cognitive impairments, comas, or even death. So, the responsible genes, there are three responsible genes. However, someone who is affected by this disorder does not have to have a mutation in all three genes. They only have to have a mutation in one gene to have the disorder. There are just three genes that can cause it. So the BCK-DHA gene is located on the long arm of chromosome 19, position 13. The BCK-DHB gene is located on the long arm of chromosome 6, position 14. And the DBT gene is located on the short arm of chromosome 1, position 21. I should clarify that the Q's that are after 19, chromosome 19, and then chromosome 6 represent the long arm. And then P, that's listed after chromosome 1, P represents the short arm. And so this image here that I have is actually the chromosome of the BCK-DHB gene. And so this pink part right here represents the centromere, and then the arrow is pointing to the location of the gene on the chromosome. So as you can see, it is located on the long arm of this chromosome and located at position 14. And these second numbers represent basically just the smaller the number is, it's kind of the positioning relative to the centromere. So the smaller numbers are going to be closer to the centromere, whereas the larger numbers are going to be farther away. And so on the DBT gene, you can see it's probably going to be a little bit farther away, more far away than these first two here. And then this disorder is inherited in an autosomal recessive manner. So in order to have the disorder, you have to have two mutated copies of the gene, and it is not sex-linked, so it's not going to be on germ cells, it's only on autosomal cells. This here is a screenshot of Project 2 that I just thought was interesting and relevant to help point out some things here. So this shows that parent 1 is homozygous for the disorder and that the other parent is homozygous normal. So therefore they don't have the disorder and they are also not carriers. And so none of their children will be directly affected by the disorder, but 100% of their children will be carriers. And so if their children reproduce with somebody who either has the disorder or is a carrier, their offspring may potentially be at risk for developing the disorder in the future. Okay, and then this is another screenshot that I took of the project that I thought was interesting as well. So this shows that one of the parents is homozygous for the mutation, the genetic disorder, and the other parent is heterozygous, meaning they're a carrier. And so in this situation, there's a 50-50 chance that their offspring is either going to have the genetic disorder, so they're going to be homozygous for the disorder, the small m's, or they're going to be carriers of the disorder. They're not going to produce any offspring that are going to be quote unquote normal. So I thought that was interesting and helps show. This is the pedigree that I put together for my disorder. And so as you can see here, the first two members of this tree, generation one is two homozygous for the disorder and one is homozygous normal. And as you can see, all of their children are heterozygous carriers of the disorder and representative of a recessive disorder. It kind of skips a generation here, as you can see. And then all of the individuals in generation 2 also marry individuals who are carriers of the disorder. And so in generation 3 here, you can see that more individuals are affected by the disorder. One thing I'll point out is that in generation 3, Amanda, who is a carrier of the disorder, married Mike, who is homozygous normal. And so their children actually will be either a carrier or they can be homozygous normal, since Mike is homozygous normal and Amanda is just a carrier. And so Landon, if he chooses to marry, or if he does marry somebody who is homozygous normal as well, he'll kind of like break the cycle of passing on that disorder. So the mutations causing the disorder, I'm only going to go over one, which is the BCK-DHA gene. And there are 80 different mutations that have been found to cause the gene to basically malfunction. And so when the gene malfunctions, it does not produce two alpha enzymes that are needed for the BCKD enzyme, which works to break down those 3 branch chain amino acids. And so when that happens, then that's when you get the buildup. The most common mutation that causes this is a point mutation. And a point mutation means just a single change in the amino acid sequence. And then more specifically, the most common change is replacing tyrosine with asparagine at position 438, and that is a missense mutation. So genetic testing, maple syrup urine disease is actually included on the standard newborn screening panel in all 50 states. So all newborns will be screened for this disorder unless their parents opt out of testing. And this is done typically 24 to 48 hours after birth, since it's so important to to begin treatment immediately after birth. There are 4 different types of genetic testing available, which are listed here. And these are the 4 types of testing that somebody could potentially order and be delivered for at-home testing. However, since it's routinely tested for in all 50 states after a newborn is born, I don't really see a specific reason why you would order an at-home test, but they are available. If a test comes back positive for the disorder, a secondary test is usually performed, and that is usually the molecular testing. And since this disorder does have known treatments and an individual can live basically a normal life after treatment, there isn't much controversy specific to this disorder, to testing for this disorder. And so early detection and treatment. Early detection is very important, like I mentioned, because there are treatments available. So treatment includes long-term diet restrictions that basically reduce the intake for foods that are high in protein, and then specifically foods that contain branched-chain amino acids. And so this is because protein is made up of amino acids, so if you reduce the intake of protein, then you should be able to reduce the amount of amino acids that you intake. There is also medications to treat episodes of extreme metabolic decompensation. If for some reason you ingest something that you shouldn't have, if you just get a buildup of the branched chain amino acids in your blood. Some of these options might be dialysis to kind of clear out that buildup. A more extreme measure is a liver transplant, where you get a donor liver that does not have the disorder so that you can break down these branched chain amino acids. However, that's less common. And then throughout your lifespan, you'll have frequent and routine follow-ups with your physician and with the dietitian, and you'll also just have frequent blood tests to make sure that your amino acid levels aren't getting too high. And then this is just an interesting snippet I thought I would add in just to represent some foods that people with this disorder can and can't eat. So the most common things that these people can eat are fruits and vegetables and breads and pastas. Lots and lots of that is what I found. Cereal-based foods, and then fats and sugars. What they cannot eat is any type of meat, including fish, eggs, dairy products, and nuts and legumes. And this is my slide of references. And so that's basically just a quick overview of maple syrup urine disorder. I had a great time researching in class, and I honestly learned a lot, and I hope you did as well. Thanks for listening.